joining, you must talk to one of these people. You must get over your fear of people like this and talk to them, as difficult as that may be. Okay?
I'm going to go off of that and say that Journal Club's actually looking for sophomores or freshmen. Anyone who's interested, please send me an email at stein@hangry.org. Anything else? Project 80, do you have any other things? Okay. This week we have Rahul presenting on a new type of therapy for diabetes. Rahul. Oh, I'm sorry. First time presenter. That means you can ask clarification questions. Don't be like, so how does diabetes affect the worldwide population of mice? No, don't. Make it clarification. Keep it simple-ish. Thank you.
So my paper's on SH2B and beta cells promoting insulin expression and glucose metabolism in mice. Well, let's first start at the beginning. What causes diabetes and how can we sort of alleviate and treat it? So diabetes basically just means there's high blood glucose and you don't have enough insulin to sort of absorb that glucose and turn it into energy. Who here knows someone with diabetes? Oh, wait, yeah. Alright. So insulin is, what I just said, a hormone secreted from the pancreas to sort of absorb glucose and turn it into energy. And so why is diabetes a problem? So diabetes affects over 300 million different people, and it can cause symptoms like extreme fatigue, and it can weaken people's immune systems to the point where a common cold can require a visit to the emergency hospital. So it can get pretty serious on a lot of different types of people. There are two different types of diabetes. There's type 1, and type 1 is when the immune system itself attacks the body's beta cells, which produce insulin, so there's no insulin production at all. And type 1 is genetic. Type 2 is what affects most people, where insulin is still produced, but there's not enough of it, and it's not being used correctly, and so that's called insulin resistance. And then as type 2 diabetes gets worse, the pancreas produces less and less insulin and builds up insulin deficiency.
Yes. How does diabetes affect the worldwide population of mice? Worldwide mice. Worldwide mice? Worldwide mice.
It makes them fatter. So, how can we control the expression of insulin? So, beta cells in the aisles of Langerhans is what sort of produce insulin. And so what researchers in this paper are trying to understand are the channel of intracellular signaling proteins that sort of signal to one another and sort of produce like a chain of signals that ends up with producing insulin. So the researchers are taking a look at those specific chains. And that's what led them to SH2B1. So SH2B1 is an SH2 adapter protein. And so it links protein binding partners together and helps to facilitate this large signaling complex. And so one important point is that SH2B1 is not an enzyme, so it won't speed up insulin production, and it won't really speed up anything. But researchers are taking a look at if you overexpress it, what are the chain of events that will happen. So in this graph right here, the researchers tested 3 different types of mice. They tested wild type, which are just regular mice, OB mice, which are obese mice without a hormone called leptin, which tells you if, once you're eating, when to stop, when you feel full. So as you can imagine, these mice just keep on eating and eating. And then double knockout mice, which are like wild type mice, except they don't have the SH2B1 adapter protein. And so, as you can see in graphs B and C, the body weight and fat content between really obese mice and regular mice just without this adapter protein are really similar. So, the researchers took a look at if this relates to blood glucose in any way, because high blood glucose is diabetes. So they're looking for sort of a direct link here. As you can see in figure A, there's much higher blood glucose in mice without the adapter protein than there are in obese mice. And if you look at the plasma insulin levels, they're about the same for obese mice and mice without the adapter protein. And so high plasma insulin levels basically means that there's insulin resistance. So you can see that these two mice are sort of, the obese mice and the double knockout mice are on their way to developing type 2 diabetes. And so now, from this graph, the researchers are trying to see if SH2B1 can directly sort of control blood glucose levels. So, how does it function? So, in this graph right here, they measured the pancreatic insulin content between the obese mice and the double knockout mice. And they found that obese mice, even though they ate a lot more food, was producing a lot more insulin than regular mice just without the SH2B1 adapter protein. And so now they can make the link that if you increase the expression of SH2B1, then you'll increase the secretion of insulin. And so before we go further, there are a couple terms we sort of have to go over. So the first is JAK2. And so JAK2 is a pathway that is responsible for basically producing beta cells, which are the cells that secrete insulin, and PDX1. PDX1 is another pathway that's a transcription factor, and in the end, it ends up promoting insulin. So then, in this next graph, the researchers took SH2B1 plasmids and co-transfected them with a rat insulin promoter called luciferase. And so what they did is by doing that and then measuring the luciferase activity in mice, they could sort of tell the different insulin promotion activity. And so they tested 4 different groups, like none, just JAK2, just SH2B1, and then both. And what they found is that when you express both, SH2B1 and JAK2 sort of synergize to stimulate insulin activity.
Did they say why they have different doses for the SH2B1?
So now you saw how SH2B1 and JAK2 sort of work together to promote insulin. So now they took a look at SH2B1's effects on PDX1. And so if you look in graph A, shRNA is introduced to the mice, and what that basically does is it blocks the effects of SH2B1. So that's not being expressed in graph A. So what that shows is by silencing SH2B1, there's less PDX1 expression. And then if you look at graph B, they reintroduce SH2B1 back into their mice, and that shows that the PDX1 mRNA levels are a lot higher. So basically, if you overexpress SH2B1, there's going to be more PDX1 expression. So why is this all important? So over 300 million people are affected by diabetes, and every day they sort of live their lives having to check their blood glucose levels often and administering day-to-day drugs. But the overall problem inside their bodies doesn't get better. What this research shows is that there is now a potential to treat diabetes at the core within the islets of Langerhans. And so next steps from this research would be finding a way to directly express SH2B1. And so one of the genes that the researchers were looking into were the CRISPR/Cas9, which I think was presented on last week. And one concern going forward is that overexpressing SH2B1 could cause hyperinsulinemia, which basically is there's too much insulin in the blood relative to the level of glucose. That's it. I know that it's often confused with just regular diabetes for high blood glucose, but I don't know the specific differences between hyperinsulinemia.
So treating it in this way could possibly lead to a reversal effect.
Yes. So that's one of the concerns that they have to sort of take into account when going forward for this.
How would hyperinsulinemia
I guess you'd sort of have to find a way to control the expression of SH2B1. So going forward, if they find a link, a genetic link that leads up to SH2B1 expression, you have to sort of be careful not to overexpress that and sort of not like really overexpress SH2B1. Does that make sense?
So, how far away? But I just want to make sure that we all understand, like, what's the distance between this data and possible therapies?
There's still a lot of distance away. They have to find another link between this and SH2B1. They sort of have to find how, if they find that link, they sort of have to find out how to control it so hyperinsulinemia isn't a problem. And then once that's found, then they have to go through the process of sort of making a drug to sort of express that target. But that process will take a while.
Think you got anything to add,